I'll introduce Geoff. So I actually loved how Marissa gave a shout out to Geoff because he actually picked the colors in 69. So I love this, how we're getting to talk about how you share data and tell your story. So Geoff Kraker, he's going to give us a little bit new style of presenting it, FlowTex, with what's called CytoBytes Live. But more about Geoff, he started his flow career with several years in the full service core at Northwestern. And there he spent the next 12 years working with high parameter analysis tool as an application scientist at various software and instrument vendors. Now he's the senior application scientist at OMIC. He's a persistent advocate for data quality and is passionate about educating people in the field with great enthusiasm, I'll comment, on approachable strategies for high-dimensional cytometry analysis. So take it away.
Great, thank you so much. I'm gonna loop in my friend Nico here in just a little bit, and I'll talk a little bit about this format. So, CytoBytes Live. CytoBytes is a web, basically a YouTube channel, kind of this effort that we've put together. We're actually technically an ISAC subcommittee as well, but basically it's a group of people who have gotten together and said, hey, this stuff is hard. This high parameter analysis stuff using algorithms is really complex. And the field could use a little help just to step up to figure out, okay, so what is dimensionality reduction? What is clustering? How do I know how many clusters? How do I know how many clusters to use in my flow sum? And other various topics around that. So it's made up of both academia and industry. And really, you know, I count myself like the least among these. There's really, really, really bright people in this group. And they're all sitting down and committing to say, hey, every once in a while, we'll put out a little 10-minute video on a particular topic. So what topics get addressed is kind of a communal decision. But basically, we want to hear from you, what you want to learn about. So if you would like to go check it out, you can go search YouTube. It's just at CytoBytes. And it looks like this. And last year, I think it was last year, or maybe 2 years ago, Peter Mage gave a fantastic unmixing talk. He's kind of reprised that and shortened it for CytoBytes. But we have Sophie, who invented FlowSum, talking about how to pick the number of clusters for FlowSum. We have, again, a really stellar cast of characters to explain things that are near and dear to them. First up is this, for me, dimensionality reduction versus clustering. This is the video that I did for CytoBytes, and you all are going to get a reprised version of that today for number one. So kind of step one is dimensionality reduction versus clustering. Step two is kind of more broadly, kind of what not to do, or like forensic cytometry, algorithm addition, and Nico's going to be talking about that. This is basically like, hey, you might have seen a pattern that looks like this. This is why this is wrong, and this is how to fix it. And we'll talk about generally like some pointers there. And then last, we'll have how to talk about high parameter analysis, these algorithm results in a publication setting. So there's lots of fantastic lead ups and agreements between our presentations, which is a relief. I'm always terrified. I'm like, nope, that's wrong because I got that one wrong. So sorry. So I think we're gonna see a lot of alignment there. All right, so this is the fun little intro we put at the beginning of every one of our little YouTube videos, and we'll dive right in. So I mentioned this, I think it was either last year or 2 years ago, and I put up a slide about dimensionality reduction and clustering, what the difference is, and I used a little analogy. I'm gonna run that one back. So if this sounds familiar, you're gonna be like, oh, that's familiar. But this picture is new. So this is Anna Belkina, who developed Opsni. She's also part of this crew. And on our most recent call, she posted this, and I was like, can I just crib that directly for this talk? So clustering, things like FlowSum, SPADE, PARC, Phenograph, dimensionality reduction, or in this case, in this little thing, visualization, these are things like TSNE, UMAP, PACMAP. We already heard a lot about that. So if these are algorithms, they look at all of your parameters at once and then try to put cells that are similar in many dimensional space in the same space in 2 dimensions. But they operate differently. Under the hood, they're entirely different algorithms. The math is different. And so they don't necessarily agree. And that's what we're seeing here is if you're clustering and your dimensionality reduction don't match exactly. Sometimes, I'm sure some of you have experienced this, they kind of smear over into each other, and she thought that was particularly apt, and I agree. So these are technically both types, clustering and dimensionality reduction. These are unsupervised machine learning algorithms. So they're AI technically, but not like the large language models that everybody is hearing about these days. So dimensionality reduction places events that are similar in many dimensional space just close together. It doesn't have to make any hard choices. It just gets to kind of mush things close. Clustering does have to make some hard choices. It says, all right, I have 30 clusters to work with. I have to split up these populations into these 30 clusters, and I'll take my best crack at it, or 40, or 60, or 10. And it's kind of up to you how many. But all of them are just different strategies. And again, this is a perennial slide of mine, but I think it's really, really fitting. All of these are different strategies of taking a multidimensional object, like a three-dimensional object, and putting it down into 2 dimensions. You can do this with a globe to a map, many different ways. It's just going from 3 to 2. There's tons of ways you can do this. So just think how many ways there are to do it. If you have 30 or 40 or 50 now dimensions cytometry data, condensing that down into just 2 can be, pretty tough. So we'll dive right into here. What is dimensionality reduction, and specifically, what is it popular, or what is it used for? Basically, it's great at showing you everything inside of a data set, all the events inside of a data set, regardless of whether you know how to define those events or not. Historically, gating is based on a hierarchy. You say, all right, I know most of the stuff I care about is 45 positive. I'll draw a gate. What about the 45 really dim stuff that just missed your gate? Is it still a cell? Maybe, but if it's outside of your gate, you're never going to be able to take a look at it downstream. These tools can help with things like that. But they're also really great at QC. So saying, hey, are there batch effects? And this is something you said before. It's very sensitive. These tools are very sensitive to batch effects, so they can be great in helping you pick those out. If you have very bright and very bimodal staining, if you put CD4 and BV421, for instance, that's going to have a big effect on the resulting kind of separation of your islands for your dimensionality reduction visualization. If you have a really dim smeary marker like CD25 and it's on a really dim floor, that's going to be really tough. It won't have that much of an effect on the structure of your UMAP or your TSNE. So it ends up being it's sensitive to your panel design. So good panel design undergirds all of cytometry, obviously, but especially here as well. You'll see those effects of good unmixing, good panel design in these results. The other thing that you can pay attention to is that if you have a very different event, it'll be far apart from most of your other events. If the events are very similar, they'll be in the same island. But a lot of them, especially UMAP, TSNE, et cetera, the distances between your islands don't matter as much. It's not linear in a technical sense. PCA is linear. And that's why you can actually have a distinction between the two. So this is actually, it's really funny. One of the figures you put up just before said that the OPSNI clusters, it said that in the figure legend, and this is a point I want to make, they are actually not technically clusters because you're not clustering. So you can call them a lot of different things. I happen to use islands all the time because it happens to agree with me. But you can call it whatever you want. Groups, and people get it when you talk about clusters, but that's not technically accurate. So in this case, this is an example. Let's see if I can get this. There we go, laser pointer up. This is an example. This is just to highlight a result of what a UMAP looks like. Or in this case, actually, technically, it's a pack map. Very similar. Find me later at the table to figure out how they're different. These are being colored by their CD4 expression. Here you can see we're changing the marker to CCR7. So you can see, oh yeah, all right, these are the CCR7 positive CD4 T cells. Here we're coloring by CD25. And you can see that there's maybe just a tiny little peninsula right here. And I want to make a point. It's important to also show density information. So here, when you're coloring by a different marker, you don't get how many events are stacked on top of each other. So it's still important to have these pseudo color plots. Clustering, on the other hand, is a way to separate your cells into discrete populations. So it's a way to basically gate without having to know the definition of the phenotype. And so it's great for taking this kind of more hands-off approach, especially if you are dealing with either new markers or kind of an experimental sample. You don't know what the biology is there. You don't want to have to know what the biology is in order to get your population separated. So this is a great tool, but it's not a tool everybody should use all the time. And it feels kind of weird being like the high parameter data guy talking about like not using clustering for everything, but but legitimately, you know, when you have a hammer, every problem looks like a nail. You know, that's kind of the adage there. You don't have to cluster everything. In fact, if you have a very smeary but well-defined, importantly scientifically defined population like central memory CD4 positive T cells, clustering algorithms really struggle with that CCR7 smear or with a 45RA smear. They really don't do a good job of getting you only central memory cells or only effector memory cells in a particular cluster. That's just not what they're built for. And so if you're trying to communicate results in your assay or in your paper, in your story about central memory T cells, clustering will prevent it, or it'll present a challenge to that communication, because your clusters will be like mostly central memory with a little bit of effector memory cells too. And people are going to be like, why do you have effector memory cells in your central memory population? And it ends up just complicating things for often not that much gain in efficiency or comprehension of your audience. So again, just keep that in mind. Use it judiciously. Basically, the results of clustering will be a new clustering channel, and then you get to gate on that to turn those clusters into actual populations. And then just something to keep in mind, whether you're using something like FlowSum, where you define the number of clusters, or something like Phenograph, where the algorithm tries to pick the right number of clusters, it's going to try to fit your data, your biology, into that number of clusters that it chooses, but it doesn't know the biology. So it's always really important with these things to take what you see as a result and then check it based on your understanding of the system. This is how people are visualizing clusters, and we'll see these show up a little bit later as well. But there's lots of ways to take your clusters and label them, but that's a topic, again, for a different conversation. But here's how you can visualize some of the clustering results. Here is that OMIP 69 figure. So thanks, Matilda, for the shout out. It is, yeah, no, it was a really fun figure to build. And we'll see this again also. But these are clusters overlaid on top of dimensionality, or sorry, on top of a dimensionality reduction visualization or projection. And so this is kind of both of them together. And when you see these kind of smears where they kind of fuzz against each other, that's maybe like from that first picture, a cat's paw, you know, just leaving its own cluster. In this case, for the most part, the clusters are aligned with the dimensionality reduction projection. But this gives you an idea of kind of where all of the different subpopulations are in their own islands. So this is where this slide comes back again. I was talking to Paul Mead just earlier today, and he said, I love that analogy. So I don't even feel bad about running this slide back because it's probably a new audience. This is a picture of my daughter's floor after Halloween. So this is one of my favorite ways to talk about this. Dimensionality reduction is like when your kid comes back from Halloween and you have a big sack full of candy and you dump it out on the floor of the kitchen table and you sort out the candies into their different little piles. That's what dimensionality reduction is doing. It's taking many different parameters, candy size and composition and color and how much you like it, et cetera, and sorting out all of those different groups. And then clustering would be if you had to take some sandwich baggies and say, hey, you know what? You have a whole pile of candy all separated out into these little groups. Now take these and put them into these 10 different sandwich baggies. And what happens with like your peanut M&Ms? Do they go in the same baggie as your normal M&Ms? Depends how many baggies you have. So clustering, you have to make hard decisions. You don't get to just like be wishy-washy or compromise. You're actually having this kind of population discretely separated into pieces. And so picking the right number of clusters is an art a little bit and a science. You'll have to watch Sophie's CytoByte about that. With that, that's a quick summary of dimensionality reduction versus clustering. So now I'm going to loop in Nicolas, and he's going to talk a little bit about this forensic cytometry. And while he walks down, this is kind of the seeds of our tutorial at Cyto this year. So if you're coming to Cyto this year, we're going to have this data analysis.
Saturday at 10 a.m.
Is it Saturday at 10?
Yep.
Fantastic. So come see a longer, more thorough version of this at Cyto. Take it away.
Thank you. All right. Thank you, Geoff, for giving some time to give this teaser about the Cyto talk. So basically, it's crime against data analysis. It's going to be separated in three different parts. So broad guideline for any algorithm, and after specific subject on dimensionality reduction and specific tips on clustering. So before, what's the basic assumption? So we want to make sure the data is unmixed and compensated correctly, that you remove any technical anomalies, so clog, air bubble events. So make sure that the MFI is stable over time, so you can do just a time gate, fluorescence versus your time, or use a QC algorithm like FlowAI, FlowCut, Peacock, or things like that. So we want also to make sure that your antibody are titrated correctly. And of course, that you select only your cells of interest. So you want to remove debris, doublet, maybe if you're only interested in your leukocyte, select your CD45 positive, and don't worry and dump everything that's maybe a lineage negative or whatever. And of course, your channel need to be scaled correctly. So all those algorithms use the graphical space. So they need to be scaled correctly for the algorithm to work properly. So what does it mean? So, well, here's a proper scales. Okay, so if we look at our dot plot here on the top, we get it on the event in the negative population below 0 and the event above 0 that we know as a flow cytometrist, there's one population. So when we run algorithm, you see that our CD4 positive population is only one population for the negative 0 and positive 0, basically. And we're going to have a full video on the CytoBytes channel. Should be released before the end of the month. It was supposed to be before this talk, but we're a little bit late, but it'll be there in the next week. So what's an incorrect scaling? So if you start adjusting your scales, we can have this butterfly effect in your negative population. So still, we have the negative population, which is everything. But because of the butterfly effect, we have events below the 0 and above 0. And because my scale is not correct, when I run my algorithm, my UMAP in this case, you see that my negative population, CD4, there is split into two subpopulation when in reality is only one. So make sure your scale is correct. Also, please don't use time parameter. Time parameter is not a good parameter to use. And Geoff and I can attest that we see many users that send us data that include the time parameter. So please don't. Same thing, make sure that you don't use any linear parameter with oxygenate or by exponential scale parameter. So don't use side scatter, forward scatter with all your fluorescence parameter, because the algorithm is gonna lean toward those linear parameters. So it's gonna split those way more based on that parameter than the rest of the parameter. So better to keep only your by exponential or oxygenation parameter. So now let's go on to focus a little bit more only on the dimensionality reduction algorithm. So when you run t-SNE UMAP, you want to make sure that you see every different population well separated. So there's sometimes technical issue, panel design, data quality, but it can be also the algorithm by itself. It can be because the algorithm didn't run completely. Maybe you didn't adjust your perplexity number or your iteration number for t-SNE or your minimum distance for UMAP. So what does it look like in reality? So here is an ineffective separation between the different phenotypes. So same thing, this island right there is our CD4 population, but we can see inside we have some CD4 negative cells. So here we didn't have a nice separation between those populations. So you can change your iteration number in t-SNE to make sure that it completes all the run. So here we have the big island, really well separated, even with 100 iteration, but we see that inside those islands, we don't see the granularity of those different subpopulation. Compare that when we start increasing the iteration number, at the end, we have a really nice separation between each different subpopulation. And it's the same for UMAP with a really a small minimum distance with a big minimum distance. So adjust the number, tweak it, try different number, and see which one give you the best results. Now, on the tip for the clustering, okay, the cluster, as Geoff says, like your Ziploc bag where you put your candies in each one of them, you need to make sure that you have enough Ziploc bag to see every single population. So he's talking about candies. When I do my talk, I'm talking about fruit and veggies that you try to put in your fridge, in the drawer, but same analogy, same idea there. So first, you need to have enough cells. So the cluster algorithm is not going to be able to see maybe 5 cells and create a cluster based on that. So you need to record enough cells to have at least 40 to 50 cells for the algorithm to be able to create a cluster. And also, you want to make sure that within the cluster, you have only one population, that it didn't under-cluster, that didn't put 2 different cell population into the same cluster. And also, you don't want to over-cluster too much. Even if Sophie is saying for FlowSum, you should put 1.5 times the number of clusters you expect. Well, if you have 50% more of cluster than what you expect, you're going to have to do Boolean gate and everything to combine the cells that are similar together. So let's look into that more in detail. So does each cluster represent one kind of the cell? So algorithm doesn't know biology. So you need to check the result by eyes and with your biological understanding. Then we have algorithm to help you there. So we have Euclid, which is an algorithm to tell you if you're over cluster or not. We have the Taylor index, we have the CV and the cluster R check. So all those are tools available to tell you if you under cluster or over cluster. So here's an example on the cluster R check. Here give you a yellow tag telling you that you have a bimodal expression of this marker in this cluster. So most likely you have 2 cell type in this cluster. And also to verify the biology, you can sort those clusters and do sequencing, metabolomic analysis, and we have a lot of algorithms to do that. GateFinder, HyperGate, or HyperFunder are some tools to give you the gating strategy so you can go and sort them. Overclustering, of course, is not limited to FlowSum, but that's the one we think first, because in FlowSum, you have to give the number of clusters you expect in your population, and we usually don't know, so we're going to overestimate that number to make sure we get every cell population. So if you overestimate that, the same cell population will be split into different clusters, and you're going to have to create Boolean gate to merge them. And let's go for the publication.
All right. All right. So this next set of slides, I have, I think, one image that almost directly matches one of the ones that you had before. But we dive into it a little bit more around specifically what are these publication considerations around these algorithm tools. So making sure that you can convey your results effectively, because like I said here, presenting any data obviously comes with responsibility. You have, number one, you have to make sure that it's collected well, but then also that you present it in a way that people can understand right away what it means. So that baseline comes with responsibility. But when you're showing abstracted data, so data that's not a single dot on 2-parameter dot plots, so people can see right away what that level is. But when you're showing minimum spanning trees, you know, like the Nolan Lab figure from that Spade paper, that's just different colored dots connected with lines. And like, what does that really mean? It took people in the field like a long time to like really wrap their head around what that meant. I cannot tell you how many people asked me like, hey, I want a figure like that. And I'm like, they sat a grad student down and had them rearrange all of the clusters and edges into that hematopoietic tree. Spade does not kick out those hematopoietic trees. I promise you, it was just a labor of love. That's what it was. Or maybe not love. Maybe they had other thoughts when they were being made to do that. But the whole point is it was abstracted. You know, like you're not seeing single cell data at that point anymore. And so, especially in a field where people are used to seeing that single cell level of data, if you show in a paper, in a poster, whatever, this abstracted kind of data, you have extra responsibility to make sure that people aren't being misled by that, as everybody I talked to was about that particular figure. So again, there's new algorithms being released. Those new algorithms come with new visualization techniques. Again, minimum spanning trees were new to cytometry when Spade came out, and now people are, mostly have their heads wrapped around it, but not everybody by a long shot, and especially not every reviewer, especially not every PI. So even if you know what you're talking about, the important people who have something to say about your work might not know what you're talking about. And so it's a good idea to, number one, understand what these new tools are providing you, and then know how to communicate that effectively. So ta-da, some of the same stuff. Coolers.co is a fun website just in general. If you want to click around and see interesting palettes, sometimes they can be really helpful. Other times they're kind of more graphic design oriented of these colors look neat together, as opposed to like, cohesive and colorblind friendly. But specifically, I want to call out Viridis, but this whole series of R-based color palettes. These are colorblind friendly, and I would recommend using these if you need these kind of spectrum palettes for talking about data. The other one that I really like, and that I did use in that OMIP69 figure, is this LearnUI.design data color picker. So this lets you pick a start point, pick an end point. It gives you the hex codes for those values in between, and then you get to kind of paste those in and build your figure that way. So just something to keep in mind. There's lots more. There's so many people who are passionate about visualization, outside of cytometry. So there's a ton of resources out there, but it's something worth digging into because you can use color to tell a story. So both of these papers I was involved in, so I can put both up here and critique them both adequately. But this is fine. This is like the kind of the default order of colors, at least that omics spits out. And it's great for high contrast situations. So if you have two clusters that came out right next to each other, you want them to look different enough to show that there's a border between those clusters. And that's great. But it does, there's not a lot of cohesion around kind of what these different clusters, what cell types they are or anything like that. When you have a little time to put into something and make it better, this is my personal preference, but I think you can start to really clearly tell a story like this. And this happens to be number 22. I don't know if you can see it from the audience, but this is the CD3 positive, 4/8 negative population. I was like, I'm going to make that purple because my 8s are red and my 4s are blue, and I'm just going to, So if you have time, and a lot of people don't have a lot of extra time to deal with this, but, it can be fun to put little nice touches like that in because it helps, again, kind of tell a story. So I'd say, being intentional about how you put these figures together can be beneficial. The other thing is when you're building these plots, you've seen a few already in my presentation. You saw them in the last presentation. You'll see them again in real life for sure. If you're building these plots, there's a couple ways you can do it. And this is a little tangent, but this is how I live my life. So buckle up. So I would recommend I labeled all of these different clusters, and you can do this same type of figure even if you're not clustering. Remember I said every problem looks like a nail if you have a hammer? Like if you're not doing clustering, if you're just doing manual gating, you can still make these cool overlay figures, and I'd recommend it. I think it's an intuitive way to see kind of what's in your tube. But there are still events that might be indescribable or might fall through the cracks in your gating. So there's going to be a difference between every cluster that you have time to name and total cells. And so you can see things like right here, there's these little gray populations. So I'd recommend if you build these figures, put total events, total clean events. You don't have to put doublets and debris and stuff in the background, but total clean events as a background color, and then put all of your labeled populations in front so people can see, hey, look, hey, this is real life. There are some types of cells, whether they're antibody aggregates or just, I'm looking at, I mean, nobody's done 60 yet, but like when you look at 60 colors in the same place, 60 different markers to describe phenotypes, I'm sure there's going to be biology that now will come out, and there might be one little weird new kind of cell that's like some weird phenotype nobody expected to be there, so it doesn't fit in your manual gating strategy, and so there's no label for it. There will be stuff like that that pops up. So just for context, put in total clean cells in the background just so you have something to bounce the rest of your populations off in context. Another quick note here is oh, there we go. Okay. Another quick note here is consider the trade-offs in novel plot types. So here's a couple of examples of like things that seem really cool and are like they drive engagement, and you can say, this is all right. This is going to get people to show up at my poster, or this is going to get people to comment on this. It's a trade-off because you're actually hiding things. So in this case, this is a plot type, and I put the reference here. And actually, at the time, and still, I think it's pretty cool. It's actually out of Jonathan Irish's lab, and it turns out it's from a long time ago in his lab. But they used density dot plots they used basically they plotted in this case CD20 versus BCL2, and then did like a z-axis coloring by a third marker, and then they used ImageJ to turn that z-axis coloring into actual like a third axis like depth, so it looks like a mountain. And I'm like, number one, that's kind of sweet. Feel free, just because you don't have every tool in a particular software package, take it and go outside and see if you can make your figures more engaging. I'd absolutely recommend that kind of cross-pollination. In this case, though, this kind of mountain dynamic hides what goes on in the top kind of back quadrant of what's going in this particular plot. So they can be really cool, but if you don't actually have them in 3 dimensions, sometimes it can be really tough to see what's going on comprehensively. So these plots are going to drive engagement, but they're not. Their focus isn't on being able to see everything that happens all at once here. And the same is true with these kind of shifted histogram overlays. People like these because, again, they're kind of cool. It's a different way of looking at the same old stacked histograms that everybody has all the time when they're talking about their results. But you really do need to keep these kind of x-axis lines for scale here in order to be able for people to say, oh, there is a shift or there isn't, because you've now turned it. You don't have that perspective anymore. So definitely feel free to use these kind of novel plot types to get people to come over and start a conversation. But just keep in mind, don't make them the crux of your analysis because it can be sometimes a little hard to get people to pick up on what you're trying to do. So we have some dimensional reduction dot plot considerations. In this case, and this is, again, a call out to scaling UMAPs. So here, this is a UMAP that's colored by CD25, And you can see, yeah, there's some negative stuff here, and here's probably where the positives are, but it's not great. This is almost a one-to-one of the slide we saw earlier. If you change the scale appropriately, you can much more, this is the exact same plot, the exact same marker, just changing the scale. You can much more effectively say, hey, look, here's the positives and here's the negatives, but you have to actually have this scale bar listed here. You can see that the scale bar changes when I've more appropriately adjusted it for this particular dynamic range. And also, like I mentioned before, make sure you're showing density, because if I was just looking at this, I'd have no idea kind of what the difference is between either end of this island. And it turns out there is a really big difference in just the number of cells that are represented there. So whenever you're showing UMaps or any kind of plot colored by a z-axis or a third marker, also show the density dot plot so that people can understand where the cells are actually in the plot. Another one that I wanted to mention kind of briefly here is sometimes people, when they give you access to their algorithms, they open source their algorithms and put them out in the world. They'll say, hey, knock yourself out. This is great. And then you try to push the envelope a little bit, and you end up running into issues around kerning, for instance. This is when you have text that overlaps each other unintentionally, or you have clusters that overlap each other unintentionally. This is something that's been fixed in the most recent version, so you don't have to worry about that anymore. But it is something to just keep in mind that the open source tools that authors make available are gifts, and we shouldn't take them for granted. So if you run into stuff like this, they appreciate feedback, and also whatever platform you're using these tools on appreciates usability feedback as well. I can say that because I appreciate feedback on our platform. Then this is something I also want to take just a little bit of time to focus on, because as people use these tools more and more, this doesn't seem necessarily like a graphics in publication kind of thing, but it is very much a using algorithm in publications thing. It's really, really difficult, especially if you look at papers from 5 years ago, but still papers all over the place today. They'll have these cool UMaps or TSNEs or FlowSoms or whatever in them, and then there'll be no way for people to reproduce that. You won't know what platform it was run on, what version number the algorithm was, what hyperparameters or settings the algorithm used at all. And so while there is a standard for information to include when you submit your FCS files, that's called MyFlowSite, MyFlowSite. You can Google it. The standard was developed, I don't know, probably over 10 years ago. 15. So it's been around for a long time, and there wasn't, the need for an algorithm version of that, but there, I think increasingly more is. Just because there's not a requirement doesn't mean you can't add it in. So this is me saying, please, 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 if you use these algorithms, please put the way you use the algorithms, the version of the algorithms, the cells you put into the algorithm, put that information in the supplementary info. So this is from that OMIP 69 supplementary material. This is the settings I used for the UMAP. And so, you don't have to go way into the deep weeds, but I'd say at least saying, what cell population, what parameters, what channels you use to generate the result is a great first step. So if you put this in, then other people can reproduce your work. When Flow repository is working, then you can download the data ostensibly, you can copy these settings, and you'll be able to exactly reproduce the figure in somebody else's paper. And that's the goal. Historically, that hasn't ever, it hasn't been something that was practically done, even though there's a lot of intention and a lot of requests. We finally got to the point where now people and editors are realizing that, yeah, putting flow data out there, the raw data out there is important. This is just as important if you're using these tools. So I would definitely encourage you to do so. And if you don't know how or what, again, whatever, come find one of us, and we're happy to talk about how to generate this info. And with that, That's the last slide, but I'd like to thank the organizers again for having us out, again for reprising this. I think it's really important, and I really appreciate the chance to come and talk. So thank you. Any questions? Oh, yeah.
I was just going to have you repeat it. Does the metadata for TISNI, UMAP, or FlowSum usually get captured, like by FlowJo?
So yeah, in OMIC, it does. There's like the, when you make a new task for any of those algorithms, there's like a settings window, and you can always go back and reference that. So yeah, it'll be listed there. You know, there's always more we could do, but yeah, it's recorded. And if you didn't get my announcement before, I really would recommend, if you know what Discord is and you're starting out on your Flow, or even if you're, you know, a Flow core manager or director, there's a flow cytometry Discord, which is a giant chat room full of very enthusiastic flow cytometrists who are very happy to help other people with their problems. There's channels on assay help, instrument help, data analysis help. There's a memes channel, which I particularly enjoy. So there's a lot of support there. So if you have questions just in general, you're like, hey, I'm just starting, check out the Flow Cytometry Discord. And there's a QR code in the coffee break slides. So yeah.
Have you ever tried uploading flow data to NIAD's import platform?
I know people have pitched that as an alternative. I personally haven't. There's a couple efforts to get Flow repository up and running again, and one was really close and then kind of fell apart. There's now a working group that ISAC has put together with some really, really, really bright people who are fixing it, but they're still in the process. So there's absolutely a, if anybody has tried to upload or download from Flow repository, maybe you've experienced this. There's been some challenges and they recognize that, and they're trying really hard to get something scalable and usable up soon, but it's not going to happen in the next couple months, I don't think.